Right, we start with the number 1 stage, the pollen grain deposit on the stigma and absorb water. After absorbing the water, we call it a process of next events, germination. Now, what is the visibility of germination? We can see small tube is coming from this pollen grain. So this one is called pollen tube. Right now, what is inside the pollen grain? It has two nuclei. So this is a nuclei 1. This is a nuclei 2. Two nuclei. This one is called generative nucleus, and this one is called tube nucleus. Tube nucleus and generative nucleus. Now the generative nucleus represents the sperm cell of the plant. So it can fertilize the egg cell. Tube nucleus is important for the guiding this generative nucleus to the ovule, right? Okay, now the fourth step, the cells of this pollen tube secrete enzymes and they digest the tissue around. So they secrete enzyme and digest the tissues in the stigma, and also this is the style part of the flower, and that's the stigma. So they start to digest the tissues by enzymatic actions. After that, digest the tissues, they benefit in two ways. One is they get the nutrition. Second one, space. Nutrition to grow and space to make this pollen tube deeper into this style and stigma and the style. Right, after a while we can notice this make a long pollen tube like this. And pollen tube, we can also see clearly the tube nucleus in the front and the generative nucleus duplicated. So it makes two nuclei now. Generative nuclei 1 and 2. So we can see 3 nuclei in the pollen tube. As it shows in the stage 4, they are growing deeper into the ovule, right? Okay, so just to summarize, number 1, pollen grain deposit on the stigma is called pollination. Then it absorbs water, and the process of growth of the pollen grain is called germination, and it makes the pollen tube, and inside it has two nuclei, generative nucleus and tube nucleus. And in the fourth stage, the tube nuclei start to pollen tube start to grow, and then they secrete enzymes to the surrounding tissues and digest and get the nutrients, and the pollen tube continuously grow. and they get also space to smoothly grow it down to the ovule. Right, like this, it goes to the very close to the microphyte. Now let's see what is happened at the microphyte. Here is the ovum and the two cells synagogue and polar nuclei and these three antipodal cells. Right, when it comes to the end, the pollen grain, a pollen tube, the first nucleus in the front of the pollen tube is the tube nucleus, and this tube nucleus breaking down. It's breaking down and it's disappearing. Now what is left now? There's two generative nuclei, 1 and 2. You know, all these cells are haploid. Then one nucleus, generative nucleus, fuse with this one, the ovum. And one generative nucleus fuse with these two polar nuclei. And this fusion of these two nuclei produce the embryo. And fusion of these three nuclei produce a structure called endosperm. So endosperm has 3 nuclei, 3N, and the embryo is 2N. So what is the next thing? Usually we know after fertilization, the flower implant fertilization, the flower become a fruit, right? So it's like this, the complete part of the flower and this is the ovule. So what we can see after the fertilization and the rest of the growth, this ovule structure make the seed coat. So this represents the ovule part. And inside we can see an embryo and also the endosperm. So endosperm is the white color one. White color one you can see in a seed. So take any seed, you can see a big white color one, which is the endosperm and which is mainly, which has mostly the starch. Even a rice grain, the endosperm is the starch producing part, right? Starch. So endosperm has starch. And then rest of the ovary change to make the fruit. If you take avocado, we can nicely see the seed coat and the white color endosperm, and also the embryo, and then the rest is the fruit. Endosperm. So this seed again plant, and it can make a new plant because the embryo can grow to make a new plant. Okay, do you have any questions? Right, if not, there's a question, right? So this part is very important that you have to study the fertilization of flowers and how it exactly take place, especially minor events like this tube nucleus disintegrate, and then one generative nucleus fuse with the ovum, and one generative nucleus fuse with the polar nuclei to make the endosperm. Actually, I forgot to say this is called a double fertilization. Because we can observe two type of fertilization. This is number one, and this is two. Because of that, we call a double fertilization. Right. We have a question from this section. It is like this. Sexual reproduction in plants includes the transfer and fusion of gametes. The digestive diagram below shows part of a flower with two pollen grains and their pollen tubes. The diagram below shows part of flower with two pollen grains. Maybe this is the one. This is the second one. The diagram also shown an enlargement of one of the pollen tube and the pH of the cytoplasm in each region of the tube. So here the part of the cytoplasm of this pollen tube, and pH is 7, 7.2, 7.6, 7.2, 6.8. At stage 1, this is the stage 1, the pollen grain contained the haploid generative nucleus. Haploid generative nucleus. Explain what is meant by the term haploid nucleus. The nucleus, this is the nucleus has half number of chromosomes. So generally idea is haploid nucleus is it has half number of chromosomes. So this half number of chromosomes produced during meiosis, right? Is reduced to half by the process of meiosis. Next one. Describe the changes in the pH of the pollen tube shown in the diagram. So you can see the pollen tube. This is the tip of the pollen tube. 7 and pH increase 7.2 and further increase to 7.6 and then decrease and decrease. So it's like a increase and then decrease. So here just 0.2 and here 0.4 increase and here 0.2 increase and here 0.4 decrease. So we can say when the pollen tube is growing, the cytoplasm which is close to the pollen grain has pH 7, and then the pH is increasing to a 7.6 maximum, and then after that pollen tube pH decreasing to 6.8. So tip of the cytoplasm has 6.8. So we can also add the greatest increase at the 7.2 to 7.6 and greatest decrease from 7.2 to 6.8. Okay, next question is at stage 2, amino acids are absorbed into the cytoplasm of the pollen tube. Stage 2 is this. Amino acids are absorbed into the cytoplasm of the pollen tube. These amino acids are used to synthesize protein such as enzyme. These enzymes are transported through the cytoplasm and then secreted into the style. Suggest what happened to the amino acid from when they are absorbed into the cytoplasm until they are secreted as enzyme into the style. Right, you see the general idea is the pollen tube also absorb the nutrients, and the nutrients are used to grow and also produce the enzymes. So stage 2 is at this point, amino acids are absorbed into the cytoplasm of the pollen tube. So then they absorb amino acid, and these amino acids are used to synthesize enzyme. These enzymes are transported through the cytoplasm and secreted into the style. Then secreted into the style mean here. Now this question suggests what happened to the amino acid when they are absorbed into the cytoplasm until they are secreted as enzymes into the style. So this question is testing the theoretical knowledge of protein synthesizing and the transportation, protein transportation and secretion by the cells. So it's a 5 marks question and also a star mark question. So you have to write relevant steps to get the full marks. So where we can start amino acid absorb. into the cytoplasm, and the end is secreted as enzyme into the style. So we have to write all the steps in between these two, right? Okay. Let's assume this is a cell, and starting is absorption of amino acid. The end is a secretion of enzyme. Okay, I think then you can make your answer like this. That's the nucleus, and this is the endoplasmic reticulum. That's a rough endoplasmic reticulum, and these are the ribosomes. And this is the Golgi. Right, the basic step, first stage is transcription, and then translation. So we can say the first stage, the genes which are responsible to make these enzymes are located inside this nucleus in the DNA molecule, and the genes are copied into the messenger RNA. Then the messenger RNA leaving from the cytoplasm and then going to the ribosomes. Now ribosome translation take place. So this amino acid attached to the tRNA molecules, and this amino acid bring to this ribosome by tRNA molecules, and then amino acids are attached to make the polypeptide chain, and they link together with the glycosidic bond to make the polypeptide chain. So translation takes place at the ribosome. After that, the polypeptide chain is moving into the endoplasmic reticulum. Then the endoplasmic reticulum fold the proteins inside the endoplasmic reticulum. After folding the protein pack into vesicles like this. And the vesicle fuse with the Golgi apparatus. And the Golgi apparatus, it is doing a modification. Example, a glycosylation. After modifying the proteins, they further pack into the vesicles. And these vesicles are moved toward the cytoplasm, and after that released by exocytosis. The enzymes are released by exocytosis. Right, you can emphasize on transcription stage and also translation stage, and also modification of a protein by Golgi, and also the process of exocytosis. As I told you before, this is very important section, protein synthesis and protein transport. So the question is asked, related to the plant fertilization this time. Right, you can add the minor details. This is the gene of the DNA which is responsible to make the enzymes, and the messenger RNA is produced by transcription, and the messenger RNA is leaving from the nucleus to the cytoplasm, and it goes to the ribosomes. Inside the ribosome, translation take place. Then this amino acid absorb, bring to the ribosome by tRNA molecules, and inside this amino acids are joined together by glycosid by peptide bonds. The peptide bond joined together and then make this a polypeptide. Then the polypeptide move into the endoplasmic reticulum, and it start to fold to get these globular shapes or 3D shapes. And then the proteins pack into the vesicles and fuse with the Golgi. Then the Golgi apparatus modified the protein, example glycosylation. And after that, the proteins are released by exocytosis. That's the enzymes, right? The enzymes are released by exocytosis. Okay, some of the theory lessons. You should remember basic stages as they are very important, and you should be able to use this theory knowledge for the questions. What is relevant to mention in the theories in the questions? So correctly identifying the exact amount of theory and how you use to get full marks. You can develop the skills when you practice the questions, more and more questions. Yes, we have done completely that part, the fertilization of plants and the practical mitosis practical, completed the mitosis and meiosis. Yes. Still 7 minutes left. We start the fertilization in animals or mammals.